Hi, Dr. Yearling here, and welcome to a guided learning activity on bacterial cell structures. I have another video where I introduced the basic cell structure terms and we categorized them as external, the boundary, or internal. If you haven't watched that video, go watch that first, lay down the basic foundation, because in this video we're going to build on that. We're going to get more neurons firing and we're going to add more content, so watch that one first. Okay, let's get started. So pull out a blank piece of paper, put away anything that might distract you, find yourself in a place where you're comfortable and focused. This will only take about 10 minutes of your time. If you find that when you color code things that's helpful for you, you may want to grab some markers as well. But all you really need is a blank piece of paper and a writing utensil to get the most out of this activity in the time we have together here. So our goal for this unit is to learn about the human microbe relationships, and the second activity that I had you do was learn about the structures of the bacteria. In this video, we're going to draw the bacteria together, getting lots of different types of learning and your neuron pathways going. So I'm going to start by drawing the cell membrane, the boundary of the cell, and I'm going to choose a black ink and the thickest ink I have to represent the fact that the cell membrane is the filter. It controls what gets into and out of the cell. I'm going to do a rod-shaped bacteria, but that's not the only shape they can be. And then I'm going to highlight the cell membrane black so that we know that that's the structure that associates with the cell membrane. Okay, let's progress now to every structure that is outside of the cell, the external structures. And I'm going to choose the color green to represent the capsule, or the glycocalyx, because this is a slimy outer layer that helps the bacteria in a number of ways. So one of which is to prevent dehydration. It also helps the bacteria adhere. So if that bacteria were, say, to get into the blood, and then if it had a capsule, it might be able to attach to the heart valve, which could lead to inflammation, which could lead to endocarditis. Without a capsule, it may not be as effective at attaching and causing disease. So the capsule plays an important role in microbiology when we look at it from a medical perspective. The capsule also can hide the bacteria from the immune system, which we'll learn about when we get to that topic. So it plays a number of important roles, and that's why we're learning it in the course right at the beginning, actually, of the course. Let's highlight the glycocalyx or capsule in green, so we know that that structure correlates to the green. And now let's go to the fimbriae. So these are long, thin projections. And of all of my drawing, which is really not that accurate, this is actually pretty close to what it looks like using scanning electron micrograph. Almost reminds me of a sea urchin, the spikes coming off of it. So what does the fimbriae do? It acts like Velcro. It helps the bacteria attach, and we just learned with the capsule, attaching can help the bacteria cause disease. So one of the reasons why we're learning it. Okay, another structure is the pili, and I'm going to make it a blue color, but a little bit lighter, because they are two different structures, but they're very similarly related, almost like cousins. And I'm going to draw it like a tube. It doesn't have to be beautiful to actually help you with learning. Okay, so this is the pili, which is actually plural, so it'd be pilis, singular. The pilis is a tube, and it in fact functions as a tube. It allows bacteria to swap genetic material. It would be kind of like if you had some really cool genetic trait and I wanted that, we could swap genetics. That's what bacteria can do. Okay, and well, let's say why that matters. Well, it helps bacteria to evolve to become resistant to drugs at a much faster rate. We'll learn about that in later units as well. Okay, let's do the flagella. So there's a lot of different arrangements. Let's just do the simplest one. So that would be one long tail coming off. They could have a cluster of tails. They could have one on one end, one on the other. They could have them all around, a lot of different arrangements. But what's the goal? I want you to know that the flagella sticks out very long and is an external structure that helps the bacteria move. Okay, so let's highlight flagella there. All right, now we come to the cell wall, and I have noted multiple times not to get that mixed up with the cell membrane. Two very different jobs. So I'm going to draw them distinct here. The cell, go a little bit thicker there, allow me to, okay. The cell wall helps give the bacteria protection as it changes environments. So you could imagine the bacteria is going to encounter a number of different environments. If it goes from the soil to someone's hands to soap to a catheter, and the cell wall can help as it changes in those different environments, protect it from bursting open. So the cell wall I drew as with holes in it because it's not the barrier. The cell wall provides structure to prevent the bacteria from bursting open. Which cell structure have we already drawn that is the barrier? If you said cell membrane, that's correct. Okay, let's label the cytoplasm. So that would be the entire interior liquid portion of the cell. The nucleoid is the location where the DNA is. And then the ribosomes, these are the protein factories for the cell. That's what I would really like you to remember. And there's many of them. Again, not accurate. If you want that, you go for a textbook image. Our goal here is to be able to draw that out to really get our brain firing. So for the ribosome, what I like to remember is it's the protein factory for the cell. If you know that, it's going to help you answer a lot of different questions that I'm going to send your way to show that you are learning and understand the content. Okay, and then finally we come to plasmids. These are small circular pieces of DNA, and they are in fact what would travel through the pilus and be swapped when bacteria are exchanging genetics. Okay, so we are only 7.5 minutes into the video. We have drawn out the structures of the bacteria, described their role, their function, and we've also said why whenever possible to help you make those mental connections more long term. What I'd love for you to do is take a break. You could go work on something else, maybe a different class or a different topic in here, and then come back. You could look at your drawing and talk through what you remember. Another thing could be to turn the page over and try to draw it on your own. What parts did you miss? Well, think about those. Give yourself some time. Maybe go back through the flashcards on the ones you missed, and then give it a break. Give yourself a day. If you really knew the material, maybe give yourself 3 days. Come back to this again and reinforce that memory so you're not forgetting what you learned, and you can have it and retrieve it in the class later on. All right, thanks for watching. Bye.